So good evening also from my side, and I'm very happy to be here tonight to share part of my research in Stem Cell Conference 2015 here in Chicago. Yes. 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 So let's change completely the topic. Let's move from the stem cell field to the cancer. What I'm going to talk about today would be about chemotherapy and highly chemo-resistant cancer as diffuse large B-cell lymphoma cancer. On the background, you can appreciate a picture of Zurich, the city where this study was performed, and where I spent my PhD and the most wonderful time. So diffuse large B-cell lymphoma cancer are a non-Hodgkin subtype of B-cell lymphoma, together with Burkitt, lymphoblastic lymphoma, anaplastic large cell lymphoma. And you can appreciate here in these slides an enlargement of lymph nodes, where is one of the major site where those tumors displace. Regarding the classification of diffuse large B-cell lymphomas, I must mention that it's one of the most common lymphoid malignancies in adults. There are 5 molecular and clinically distinct subgroups, and in red I put the biggest one, which is the NOS, or not otherwise specified, which is this study is focused on. All the rest are minor. So before going through some results, I just want to give you a brief introduction about how B-cell develops and differentiates. So B-cell development and differentiation has been defined from many people in the field as a disaster waiting to happen, because so many genetic mutation and arrangement can occur during B-cell development. So if a differentiation arrest is occurring very early on during the development process, this due by different kind of mutation, tumor arising from this step on are classified as germinal-center or GC type, and associated with very good outcome. Later on in the differentiation process, we have like ABC type or activate B cell, so cancer that are arising from mature stage of the B cell development. They are associated with very poor outcome. They are lethal. And we have a third group that actually is not a real group. It's more a morphological variant of diffuse large B-cell lymphoma cancer. They are also known in the field as HR, or host response type. This is because T lymphocyte and dendritic cell infiltration were found in different patient tissue. So this classification was very important for us because our gene candidate protein, known in the field as B-aggressive lymphoma proteins, or BAL, were found upregulated in the host response type tumors, which are the most aggressive and highly chemo-resistant. So when we talk about this category of tumors, we just talk about survival in terms of a few years, but there is no cure for this class of tumors. So because those proteins are inducible protein, and they belong to the interferon state signaling pathway, very well known in cancer, we focus our study on the investigation, deep investigation of this signaling, particularly the type 2 interferons, which include interferon gamma. Interferon gamma, this is just briefly how the signaling works, binds to different receptors, interferon gamma receptor 1 and 2, which activate JAK kinases family, which goes to activate one of the major transcription factor in this family, which is STAT1. STAT1 homodimerides, translocates from cytoplasm into the nucleus and binds the gas element, which are going to activate all the interferon gamma responsive genes downstream. Among all, IF1, which is very well known in cancer as a tumor suppressor and pro-apoptotic genes. So it will be very important for you to focus on these genes. You will see it again during the presentation. So what about the clinical outcome and therapies for this lymphoma cancer? I draw here a balance just to summarize what is the current state. As I said, most of those tumors are highly chemo-resistant. So we have radiation prior to chemotherapy, which is CHOP, or combination of different steroids, drugs, and chemotherapeutic drugs as genotoxic, interferons, of course, that are used in different kinds of cancer, and FKB inhibitors, of course, as well. But still, this is not enough, and the therapies for those tumors really needs to be improved. And an improvement for the therapy has been done by immunotherapies, for example, the combination of chop with monoclonal antibody like rituximab. And BCL6 inhibitors, which is one of the often mutated oncogene in the signaling, are very important for the lymphomagenesis. But still, the balance is still not yet. We have still to tip the balance on the left sides if we want to improve the therapies for those cancers. So the protein we find out, my institute in Zurich is also known as a PARP institute. We work mainly on poly-ADP ribosyl transferases. The subgroup of protein we analyze in this study are known in the field as B-aggressive lymphoma protein, or BALs, and they belong to this PARP superfamily. But what is the ADP ribosylation reaction cycle, or what those proteins do really? So the ADP ribosylation is a post-translational modification of a protein, as phosphorylation, for example, and is reversible. It's mediating many different functions in the cells: cell development, cell cycle, apoptosis, and transcription. So those proteins are mainly known as chromatin remodeling and transcription regulators, especially for DNA repair genome instability. So we have two classes of protein, the poly-ARDs and the mono-ARDs, according if they mediate poly or mono ADP ribosylation. They use an AD as a cofactor to make the reaction happen. And it's reversible, as I said. So this is the molecular structure of the proteins, how they look like. So 3 members belong to these proteins, BAL1, 2, and 3. We analyze in this study mainly BAL1. Our study has been done deeply also for the other 2 members of the family, as some others, but no time to go through all of them today. Members of the mono ADP ribosyl transferase PARP superfamily, as already mentioned. They contain macrodomain that you can appreciate in red. So the macrodomain has an enzymatic activity. It can be targeted by drugs. So what we would like to try to do, and this will be like one of the aims of follow-up study for these results, to develop peptides that can inhibit those macrodomain to stop the interaction. This protein are going to misregulates the interferon stat signaling pathway in those cancers highly chemo-resistant, not responsive to therapies. As I said, they belong to the interferon gamma family, and they are shuttled. So you can find those proteins in the nucleus and cytoplasm as well. So this is the whole family, small subgroup. The very few members of this family are very well known. For example, PARP1 in breast cancer and DNA repair. None was known about this subgroup as monoPARP. And for the first time, we found the link with cancer, not only blood cancer, but also prostate cancer. But today, I will focus only on the lymphoma cancer. So some results now. In our lab, we're available more than 14 different DLBCL cancer cell lines, going from the very mild to the very aggressive phenotype. The last one particularly is the host response type model, so the one that doesn't respond to any therapy and has T lymphocytes and dendritic cell infiltration. So we analyzed a different panel of interferon gamma responsive genes, and we found out that in the aggressive phenotype and not responsive to therapy, the interferon gamma signaling is constitutively activated. If you also see in B, upon interferon gamma stimulation, those proteins are already detectable in absence of any stimuli. This happens very often in cancer because, as we know, cancer cells are completely messed up. They don't follow any rules, and they have a lot of mutation. In this particular cancer cells, we have a constitutive activation of interferon gamma signaling, particularly not only by detecting by 2 different phosphosites for STAT1, and enhancement of BCL6, which is one of the major genes involved in this cancer. But the signaling downstream, having a look to IRF1, seems not really on, which is surprising because it's all activated, but just upstream we can see a signal, but nothing is happening downstream. So the question we would like to answer is like why the B-aggressive lymphoma proteins, the BALs, are upregulated in those tumors, and if has anything to do with the block downstream of the major interferon gamma responsive genes, IRF1, which is pro-apoptotic and tumor suppressor in breast cancer, has been already widely described, never described before for blood cancer. So this we have is an immunofluorescence for subcellular localization of STAT1, the major transcription factor in this signaling. We have STAT1 already translocated in the nucleus, which means active, in absence of any stimuli. This is the active STAT1 compared to the inactive one in the aggressive cancer, lymphoma cancer cells. And this is the interferon gamma receptor, because the first question for us was, what about the receptor? The receptor is mutated, it expresses that depending on something that is completely unrelated on the overexpression of our protein. But the receptor is expressed normally, and when we boost the system by adding exogenous interferon gamma, recombinant interferon gamma, the receptor is internalized. Then we went to measure if there were any endogenous production of interferon gamma. It's known that B cells, they don't produce interferon gamma. Normally T cells, they produce interferon gamma. But those are cancer cells. These are not normal cells. We could find an endogenous production of interferon gamma, which could explain why the system is kind of saturated. So STAT1, BAL1, there is a correlation on the protein expression level. We try to knock down STAT1 and have a look to BAL1 to see whether our protein was strictly correlated to the major transcription factor in this signaling. And the answer is yes, it is, and not only on the protein level, but also on the chromatin level. This is a chromatin immunoprecipitation. So it was already known that STAT1 is activating all of the genes, interferon gamma responsive genes, and by chromatin immunoprecipitation, we could conclude that not only BAL1 is a target for STAT1, but it's also able to autoregulate its own expression. So exists a feedback loop of autoregulation, and BAL1 has no transcription activity recognized, but belongs to a big family of transcription regulators, so was not surprising for us. Because all the data I have been showing so far have been done in cell lines and not in primary tissue, we started a collaboration with University Hospital where 250 patients, Swiss patients with DLBCL has been analyzed, because we wanted to make a correlation in vivo to see what our study in vitro were able to be compared with something that was, let's say, more real. So we had to look for 3 different molecular signatures. 2 different phosphosites for STAT1, tyrosine 701 and 727, and IRF2, which is known in breast cancer, as an oncogene. And it seems that 18% of the patients analyzed, they were positive for this molecular signature, which is in line with what we observed in cancer cell line. Also, we were very happy to see that we have a correlation in vivo. So we could use this B-aggressive protein as potential new targets for therapy. As I don't know if you are aware that there are PARP inhibitors in clinical trial for different kind of cancer, mainly breast cancer, ovarian cancer. We could think about different kind of cancer, like blood cancer, that hasn't been suggested before. The next question we wanted to answer was about what happened to the cells when BAL1 is not expressed anymore. So let's have a look to some function. So it seems that if we knock down BAL1 out from the system, the cells are less viable. They start slowly to die, as you can see here. And what happened to the signaling cascade, so all the interferon gamma responsive genes? IRF1, the pro-apoptotic genes, is restored. So we have again apoptosis, we have again tumor suppressor expression and downregulation of oncogenes, which makes for us clear that IRF1 and BCL6, but also some other of those genes, could be potential target for BAL1 because there is an high correlation. Of course, this correlation is mainly based on the protein expression level. I think I didn't include the chromatin studies, but we then try to analyze the promoter of those genes to see whether the transcription could be regulated from BAL1, and it seems that we have 2 potential candidates, which is IRF1 and BCL6. I wanted to show you an apoptosis analysis for all the major pro and anti-apoptotic genes involved in this system. It seems that when BAL1 is knocked out of the cells, we have activation of the 53p21 network, which to this network also IRF1 is one of the players. So it's in line with our initial hypothesis. So BAL1 is misregulating interferon gamma signaling, inhibiting apoptosis, and when BAL1 is not there anymore, we have apoptosis. So we have a correlation with the death those extremely highly resistant cancer cells. And this is a survivability assay to see what happened by using as control a topoisodoxorubicin, so just drugs that will allow us to see whether the cells are already going to apoptosis without any additional extra stimuli. So this is the model, just to conclude. So this leads me already to some conclusion. So what we propose here are novel targets for highly chemo-resistant tumors, as blood cancer, diffuse large B-cell lymphoma cancer, where the interferon gamma signaling is completely saturated, constitutively activated. BAL1 can interfere on different level, on the protein level, so interfering with the phosphorylation of STAT1 then means is activation of interferon responsive genes downstream, can inhibit this signaling directly, avoiding the activation of IRF1 on the chromatin level. So we have regulation on two different levels, protein level, chromatin level, and also an independent mechanism which is completely not clear for us. We just know that BAL1 can bind the promoter of STAT1 independent genes as BCL6 because we have been doing the chromatin studies for that. What's the mechanism behind that? This is still not clear for us. But what happened then, having a look again to this draft, to these balance, what we propose here is like to improve the current therapy. We could try to combine STAT inhibitors, which are already used for the treatment of cancer, of breast cancer, together with inhibitors of PARPs, in this case, specific inhibitors for the microdomain of PARPs, to help the response of those cancer cells to the current chemotherapeutic regimen, especially the one that will be treated with interferons. So BAL1 and also the other BALs are already all markers for DLBCL cancer, especially for the one that are associated with an interferon gamma also inflammatory response and associated with very poor outcome. So this is what we would like to test, and additional studies are ongoing. I think this is all from my side. These are all the people working in my institute. These people from the lab that helped to perform the study, and Mani from Novartis, SNF. Thank you for your attention. I'll be glad to take any question, if any.
For 5 minutes, only one question.